Okay, great. Well, let's begin. So thank you everyone for coming and attending the sort of second Brain Stimulation Journal Club. So today's topic is, I think, a fascinating area of research. So it's the combination of transcranial magnetic stimulation, TMS, with EEG. And so, as we know, EEG has some unique advantages over other neuroimaging techniques, fMRI, PET. One of the most important being the capacity to detect rapid cortical activity that occurs on the order of milliseconds. And so, since the first seminal publication that used TMS combined with EEG, which I believe was about 2.5 decades ago now, in the late 90s, the technique has been used to uncover a great deal of information about how the brain works. We've used it to probe functional connectivity between regions of the brain, to investigate excitatory and inhibitory processes, using single pulse TMS evoked potentials, paired pulse protocols, Licky, SICF, so on. And it's also been used to explore changes in brain activity before and after an intervention. So, for example, a treatment course of brain stimulation or a treatment course of a medical drug. But with those insights and opportunities, we've had several challenges along the way. So first of all, finding creative ways to minimize the artifact induced by TMS through clever engineering solutions, optimizing the EEG processing pipeline, distinguishing between relevant signals and secondary effects of the TMS protocol with EEG. So things like the brain's auditory response, the clicking sound, the effect of skin sensations, so on. And finally, once we've taken all that into account, using the information that we've gained to actually understand what the data is telling us about the brain. So piecing together what a change in a specific EEG component or amplitude actually means within the context of an experiment. So thankfully, our speakers today are working on these problems, and I'm very much looking forward to hearing what they have to say and what solutions they can give us. Now, before we begin, just really quickly, I'd like to say if you have any questions, could you wait until the end of the three presentations? And if you like, you can either ask them yourselves at the end, or if you would prefer, you can type them into the chat, and I'll be happy to relay the questions to the speakers when we're ready to. And so with all that being said, I'd like to welcome our first speaker, and so I'll give it over to Lorenzo Rocchi.
Thank you. Well, just a brief introduction, I think is due. My name is Lorenzo Rocchi. I'm a neurologist by training, but I'm always been interested in clinical neurophysiology. So I've been trained in that as well, and I became fascinated by EEG and TMS in the last years, although my background is mostly clinical neurophysiology and movement disorders in particular. So I will talk about a paper I wrote some time ago about the possibility to disentangle different sources of activation in the context of TMS EEG. So here is it. Okay, so I'm in the University of Cagliari now in Italy, but this work was done at UCL, so I think it's just right to give credit to UCL for this. So as we were saying, I will talk about TEPS, which stands for transcranial evoked potentials, which is a signal in the time domain we can get by combining TMS and EEG. Now, as the word says, it is just a series of deflections recorded in EEG, which follow a TMS pulse. Now, it seems easy, but it's quite challenging because unlike more simpler kind of motor evoked potentials like the motor one, which is recorded peripherally, there are challenges related to the fact that we have so far a limited understanding of what is a clear brain response from the TMS pulse and what is a contaminating signal. So these contaminating signals can be listed under three large categories. So a couple of them are artifacts. So the most tricky one to deal with are artifacts related to the TMS pulse itself, which can induce electromagnetic artifacts and EMG activity, so muscle activity directly from craniofacial muscles. And then we have artifacts related to the EEG itself, which are not locked to the TMS pulse, such as spontaneous eye blinks, continuous muscle activity, and so on. I'm not really going to talk about these two categories today, because what I'm focusing on is a category which does not include really artifacts. You might you might sometimes find the word artifacts related to them, but in fact, we are talking about auditory evoked responses and somatosensory evoked responses in the context of TMS-EEG. They occur because when you give a TMS pulse, you have a definite click associated to that, and then the magnetic field generated by the coil can, well, not just can, it does excite sensory fibers within the skull. So these are not artifacts. These are true brain responses, but they are indirect brain activation. So we really don't want to see them. So our aim is to minimize them somehow. So this is just a brief summary about the recent consensus papers about TMS-EEG. The situation is not very clear because there are a lot of computational methods, like, well, probably ICA, independent component analysis, is the most commonly used to remove unwanted signals from TEPs. The problem is none of these methods is perfect. They are not able to separate true cortical responses from indirect responses with a high specificity. And most importantly, they are usually applied to true artifacts. So evidence about the possibility to use them to get rid of sensory responses is not very clear. So the point is to try to minimize both during the experimental session rather than being invasive and remove a lot of signals with these methods here. Now, this is a list of artifacts really, but we are not really interested in that. Just to tell you that some of them, like the TMS pulse artifact and the EMG and the discharge artifact, are easy to recognize in terms of spatial distribution and frequency content. And so this is what we are really interested in. So auditory evoked responses, they are quite known in terms of distribution. So they tend to be in the vertex, and they tend to be biphasic with a prominent negativity and a positivity. However, the extent to which they can contaminate the TEP is difficult to assess. And then we have somatosensory evoked responses. I'm just putting a question mark here because they are far less characterized. So I will talk more about them in the context of the paper I have to comment. Let's say that if you do things right in terms of pre-processing and experimental collection of data, this is what you should have. This is an example of stimulation in the motor cortex. So roughly here, the left motor cortex. So the TEP should be composed of deflections. The amplitude should be within a few microvolts. I mean, it can be 5, 10, even 15, but that's the range. You should have more prominent peaks before 100 milliseconds, and they should make sense in terms of the stimulation side. So if you stimulate here, main responses should be there, especially the early ones. This is another example of a good response in a particularly good subject, I would say. Now, the problem is, before we were talking about some issues that have been found in the literature, is that the reproducibility of these responses have been quite low across the literature. So I'm sure you all agree that these responses look quite different, and there are also extreme cases of differences across the literature. So at some point, some investigators tried to look into the issue. So why is it that we have these big differences in signals? And there were a couple of serious attempts to disentangle to try to understand why we have these differences, with a particular eye on the fact that sensory input might be the cause of this differences in signals found in the literature. So these investigators here tried to compare taps obtained by two different directions of the coil with standard TMS, and then they compared the taps obtained with these conditions with one so-called realistic sham condition, which was basically it included auditory stimulation and somatosensory stimulation in the form of electrical stimulation of the scalp in an attempt to mimic the sensation given by TMS, and in two different sites. Well, the results were quite striking because the main finding is that there were basically no differences. So the signal induced by real or sham TMS, regardless of the stimulation site, were really similar and highly correlated. That was quite striking and I would say quite worrisome, to be honest. And there was another attempt with a similar method, although a slightly different method, because the electrical stimulation intensity was smaller, and the coil, instead of being close to the head, was far away. But besides this well, I wouldn't say small, but not really big methodological differences, the results were completely different. So in this case, there was a very big difference between the standard TMS and the sham TMS. So with hindsight, there were some probably some issues with the first paper because the pattern of responses elicited was not really compatible with the TEP, because it was made of very small early responses, very large late responses. There was some doubt about the electrical stimulation intensity used, up to 21 milliamps. I can assure you that's quite painful. And maybe the loudness of the TMS click was a bit high, so maybe the noise masking was not very effective. But the problem remains. And in particular, these two studies had one limitation, which was the sham stimulation did not allow to separate what was auditory response and what was somatosensory response. So to try and make out these differences, I designed a bit more complicated study, which included 7 blocks of stimulation. So the first 2 were pure auditory responses with 2 different coils, like a standard coil and the sham. This was let's say a side objective, just to see if the 2 gave similar responses. Then I sought to understand how the masking noise could suppress auditory responses, both when the coil was spaced from the scalp with a rigid cube, and what I call sham here is just the sham coil attached to the scalp. So there was no electrical stimulation here. So the purpose was to see how effective the masking noise can be in this context. Then I sought to characterize mixed responses. So with a standard coil this time, with and without noise masking. So to see the effect of the noise masking on real taps. And then I try to see what happens if you stimulate the scalp alone with electrical stimulation. To calibrate the electrical stimulation, I tried a different solution. So I use this formula here, which basically is, so this is the electrical stimulation intensity. So this is based on the electrical stimulation threshold weighted by the ratio of the TMS intensity, which was 90% of the RMT, and the somatosensory threshold obtained with the TMS. I got a result of 5.78 milliamps, which is somewhere in the middle between the 2 previous studies, and I think it was quite okay, because roughly half of the subject judged this to be stronger than the TMS sensation, and the other way around, the other half. And then I ran a cluster analysis with 3 different time windows of interest based on the global mean field power obtained in the TEP not masked condition. So it was the one with a larger signal because it included real responses and auditory and somatosensory responses. So this is descriptive results. The first thing to note is that the auditory responses obtained with the two coil, the real and sham coil, were the same. This suggests that the auditory matching was effective and that what really counts in terms of AEP generation in this context is the loudness of the click, not really the subtle difference in quality of the sound between the two coils. So this is the effect of noise masking on the suppression of AEP, and as you can see, in both cases, so in this case, the coil was spaced from the scalp by a 5 centimeters rigid cube, and here it was attached to the scalp. In both cases, responses were effectively suppressed. This might suggest also that the coil vibration does not really give rise to any detectable response. We should have seen this here. And these are the mixed responses. So this is quite important because, as you can see, they are very similar in the early bit. So whether you apply noise masking or not doesn't make much difference in the early bit, but there is a massive difference in the late time windows of interest. So you have this big central positivity, which is present only when you don't suppress the noise with the masking noise. And then electrical stimulation alone doesn't really do much. There is just a small midline response, but no visible activation of the primary somatosensory cortex. These are comparisons. So this is quite obvious. Here I am comparing auditory responses masked and not masked. And as you see, in one case we have a nearly flat line, in the other we have this negativity and positivity, which are both central. And comparison between the sham and the AEP mask does not give rise to any statistically significant differences. And this is expected as well. So when you compare real TEPs masked with auditory responses, you have that the two most important differences are located in the early bit, because you have a complex response for TMS, which is not present with only auditory stimulation. And the other way around, in the late time window of interest, you have this big positivity with auditory response, with auditory stimulation, which is not present with TMS. This is somehow reflected in the following comparison. Here I am comparing TEPs obtained with real TMS, either with or without noise masking. As you can see, the big differences are in the middle and in the late time windows of interest, especially the late. So these vertex responses. Well, this is just to say that TEPs are quite different from responses elicited by electrical stimulation alone, and the latter are not much different from the sham stimulation itself, apart from a very tiny response here, midline, but very small amplitude. Now, there was a problem here which led us to do further analysis, which is we noticed differences in the early and the late time window, but not really in the middle time window, which is the one corresponding approximately to the so-called N100, which is a quite famous step component. So the point was, since I have a similar wave in the AEP and in the TMS, are they the same? Is it possible that the noise, the TMS click was just not properly suppressed here? So I ran two different analyses. One was a correlation and the concordance analysis between the TEP masked and the AEP not masked to investigate whether there was some degree of correlation or concordance between the two negativities found around 100 milliseconds. The result was negative. So the two are not correlated, not concordant. And this was one more proof to state that the suppression of the TMS click was effective, because there was no correlation and no concordance between the responses, the auditory responses masked and not masked. So the point is that those two negative waves we saw, one in the condition of TEP masked and one in the pure auditory responses, are indeed different. So they might have a similar distribution, a similar spatiotemporal profile, but they have different properties, because the one elicited by pure auditory stimulation, which is this, is more central. It is readily suppressed by noise masking. It's always followed by a vertex P200, has a slightly shorter latency. This is one additional analysis which shows that the latency of the TEP and 100 is slightly longer than the auditory one. And it's also present with electrical stimulation. So if you recall here, there was this light positivity, which has the same distribution as the one elicited by auditory stimulation. By contrast, the TMS N100 is not suppressed by noise masking, has a slightly different distribution, not enough to find a statistically significant difference, but still more lateral. It's not followed by a P200. You can't really see a P200 here, while there's a massive one here, and the latency is slightly longer. And this is quite interesting, because it's a case which is quite common in electrophysiology, where two waves have a similar spatiotemporal profile, but a different physiological meaning. So what do this N100 and P200 elicited by auditory stimulation represent? Actually, this is one other case where there probably has been a bit of miscommunication between different fields of electrophysiology, because these components were already well known to scientists dealing with the somatosensory system. Well, I would say sensory systems in general, because you can find similar components like vertex negativities and positivities with ERPs, irrespective of the sensory channel used for stimulation. So you find similar responses in case of nociceptive stimulation, but auditory and also visual. This is because these components are so-called supramodal responses. So they are related more to the saliency of stimulation, not the sensory content. And this is the reason why we found them both with auditory stimulation and with electrical stimulation in this context. Okay, now this is just control experiments of the same paper. Less important, I would say, but there are still a couple of important messages. There were two additional conditions. One was electrical stimulation of the scalp with a higher intensity. So this time, not the formula you saw before, but just the threshold multiplied by 3, which was around 9 milliamps this time. And when this was qualitatively matched by the subject with the TMS, it was around 87% MSO with a monophasic coil from Mudstein. And then there was a so-called realistic sham, similar to the other two papers I mentioned before, because it was a summation of auditory and electrical stimulation. So the first condition was really done to investigate a dose effect of the electrical stimulation, and the sham was investigated to understand whether there were super additive effects of auditory and electrical stimulation. So the answer to the first question was that yes, if you increase the intensity of the electrical stimulation, you get a somewhat larger response. I would say a somewhat larger P200 more than other components, not much larger, because there was no statistically significant difference comparing this with the stimulation block recorded before with the standard electrical stimulation intensity. And the distribution profile was the same. So it was still a vertex response. And comparing the realistic sham, so this time the summation of auditory and electrical stimulation to just the auditory stimulation, we got a larger response with the realistic sham. But then, if we added the auditory responses and electrical responses with the realistic sham, there was no difference. So there was no really any hint of superadditive effects of the two modalities of stimulation. So in conclusion, the realistic sham seemed to be close to a linear summation of the two responses, auditory and electrical stimulation. So these are the main results. So in summary, with subthreshold M1, so 90% RMT in this case, it is possible to obtain genuine cortical EEG responses lasting around 300 milliseconds, because that was the time window of interest. Of course, I cannot stress enough how important it is to mask the TMS click, possibly with the masking noise and ear defenders, because it is quite important to avoid contamination by auditory responses. The role of somatosensory input is quite unclear. The problem is that this aspect was not thoroughly investigated in the present paper, because all I did was stimulate the scalp with a low electrical stimulation, but there are other issues which have not been investigated, like twitches in cranial muscles induced by electrical stimulation with higher intensity and so on. In this case, I had no muscle activation because the electrical pulse was applied on M1, but probably with more lateral areas, that wouldn't be the case. And then again, taps need to be lateralized, coherent with the stimulated area, and the earlier one needs to be larger, as you can see here. Otherwise, you are probably dealing with auditory responses, and yeah, that's it really. Okay.
Fantastic. Well, thanks very much for that. I'll remind everybody we'll be applause from the side as well. We'll have the questions at the very end. So everybody feel free to write them down, put them in the chat if you like, or just remember them and then ask them later on. Okay, great. So next I'll invite Pedro Gordon to present his talk.
So hello everyone. Can you hear me and see the presentation?
Yeah, we can.
Great. So first, a bit of introduction about myself. I am a postdoc at the University of Tübingen in the laboratory of Professor Ziemann. By training, I am a psychiatrist, but I am interested in this sort of things. That's why I've been doing this for the past years. I've been invited to talk a bit about my paper that I published in 2018, which for me, it's forever ago. But the thing is, since then, Dr. Rocky also published his paper, which, to be honest, it's an improved version of mine with much more interesting things. So yeah, I'll try to still make it interesting, presenting a few new things that we've been trying. But still, I will refer to his presentation a lot because in the end, the subject is pretty much the same, which is focusing on trying to figure out what is, in TMS EEG, what is the real response from cortical activation, like the direct cortical activation by TMS instead of this peripheral activation. And well, just a brief overview that has already been mentioned. TMS EEG is offers a lot more with respect to information than previous methods, such as TMS EMG, which is interesting in itself. But it's limited to the motor cortex, and it's limited to just one dimension, motor evoked potentials. Whereas TMS, you can get a information about the cortical region and other cortical regions, the time evolution of the response that you get from this stimulation. So we have possibly more information about the system that we are investigating. But of course, there's also this caveat that inevitably you might get some responses from sensory input, not only the auditory, but also this sensory, because the coils or the induced electrical field is also going to depolarize the neurons in the scalp, also possibly provoke motor twitches. And if you're simulating suprathreshold M1, you'll get a motor response and you get a reafferent potential, which are things that had already been observed and discussed since the development of the method and confirmed ever since. Yes, there is such a thing as this contamination of the TMS EG signal by these peripheral activations. And this is an issue that should be dealt with somehow. However, yes, the field has advanced. There are many things that have been studied. But for some reason, I had the feeling that this was left aside. Nobody gave much attention to that. Like, okay, this is interesting. We have translation from the TMS EMG to TMS EG. Here, for instance, using the silent period, correlation between the silent period observed in EMG with the amplitude of the N100, possibly because both both represent a inhibitory activity in the cortex after the stimulation, which in pharmacological studies kind of confirm this, suggesting that this phenomena is mediated by GABA B receptors, here with the baclofen increasing the amplitude of the N100, and disturbed by GABA A substances with alprazolam and diazepam decreasing the amplitude of the N100. And interestingly, they also decrease the latency of the silent period. So everything's kind of making sense there. And using this as a disease marker in this figure, suggesting that maybe attention deficit disorder, which some suggest might involve some inhibitory dysfunction, here presenting decreased amplitude of the N100. But then again, what about the peripheral activation? Most of these studies haven't brought it up. So what's up with that? So around this time, it was 2017 or 18, no, 2017, I started hearing some rumors that, well, people have been conducting these studies comparing this realistic sham and real TMS, and found out that, well, it's actually the same. It's the same signal. So maybe the TMS EG, it's all peripheral activation, which I was surprised, like, well, okay, this is important, but where is it? Where's the publication? It hasn't been published. So in the end, I got tired of waiting and decided to do it myself and see what happens. So here, the results comparing a real TMS using real TMS and sham, which is a coil that reproduces the click, plus electrical stimulation. And yes, apparently there are some differences. Differences both in the time course and also in the time frequency response. So yeah, everything is good. So there is hope. However, this paper was heavily criticized for many reasons. And here are some of the reasons. There was no psychophysical assessment of sham procedure. related to that is that the electrical simulation intensity was too low. There is no way for me to, well, there is no way to prove this, because then again, we wouldn't have needed a psychophysical assessment to see if indeed this the perception of the stimulus of both conditions were comparable, but we didn't do it, so we cannot say. But indeed, it is low. There is no control for the motor reafferent potential, because yes, there was in the sub-threshold here, there is no motor response by definition, which is present here. So, well, maybe part of the signal, or part of the difference in the signal, is being caused by that. Finally, it is possible that the sensory input from the sham does not match the real TMS, therefore is not a true realistic sham. Therefore, yeah, this study is flawed. So then I was thinking, okay, so what is this peripheral activation after all? And just as Dr. Rocky mentioned, well, it's not simply the input, the arrival of the sensory input into the brain. It's more than that. It's this supramodal response, possibly, but definitely due to this an arousal or a salience, to the salience of the stimulus, whatever the stimulus is. It can be auditory, somatosensory, visual, whatever. It's a response to new information arriving, and also possible preparation for a response. So it's kind of complex, it's supramodal, it will happen in any kind of sensory input, which mean, and it's been observed many times, and it's especially visible in the N100 P200, which means that if this is indeed a potential, well, maybe here what we see is not really a tap. This is not really a tap compared to non-tap. Maybe this is just the difference from different sensory inputs, one more salient than the other, because the TMS is more salient, that is rather low electrical stimulus, and which is causing this, and also causing the P100, P180 or P200, which confirms the criticism that we received. And in the time frequency response is translated by this alpha and beta desynchronization. So, given that kind of disappointing outcome, what did we do? Well, first of all, we had a look at what had been done ever since in terms of realistic sham. Well, there's the Konji and collaborators paper that Dr. Rocky already mentioned, that surprisingly show that indeed these two, both the real and the sham conditions do elicit very similar responses. But if we look careful at the at this, for instance, which they did, this psychophysical assessment, well, these conditions are not really comparable, are they? Which means that the salience of the stimuli are different. So it is also not a realistic sham by these standards, because they're not comparable, because the individuals could clearly perceive a difference. Still, they managed to show that the signal is comparable. Of course, then there are some criticism about this work because the intensities that they used for the real TMS is too low. But this is not the why I'm bringing this up. I'm mainly interested in the sham, and in the end, the sham not realistic for this reason. Raffin and collaborators, they published this paper in which they used in the first experiment, simulating M1, they tried to see differences in the evoked response with increased stimulus intensity and compared to a standard sham, which you can see here kind of has the same profile, like this negative N100, positive P200, but it's definitely not comparable. We'll say, well, isn't it because, or is it because indeed the sham, indeed the sensory input or this peripheral activation is significantly different from the TMS, or it's simply because it was not the realistic sham? Maybe if you it's not enough to create a standard sham. You have to pair the sensory input to each and every TMS intensity. And by doing this, they actually see that by increasing the intensity of the electric stimulus input, they start seeing that, well, these the shape of the induced response, yeah, it gets similar to this one. So yeah, if you actually want to suppress the response from the peripheral activation and get this true tap, you have to do better than this, than just creating this standard sham. So we have to come up with this truly realistic sham, and that is what I wanted to do, to create this ultimate sham that would perfectly pair the sensory input from the real TMS, so that I can compare the two, possibly subtract one from the other, and get the true the true taps. Right. So how does one go about it? Well, I thought, okay, let's do the following. Let's use the real TMS with the masking noise, because it's very important to suppress the auditory input. And this has been proved to be very efficient. Still, let's make sure that for some people that still can hear behind the masking noise a bit of a click, well, I still pair it with a sham TMS. So the auditory input is controlled. Right, so then I have to use an electrical simulation to match the somatosensory input from the real TMS. So my sham condition must have a matched electrical simulation that has the same properties of the TMS, right? Okay, and I will assess it with a visual analog scale. And on top of that, I'll throw this two alternative force choice, in which subjects will experience both conditions, and they have to tell them apart on the spot, and see if even if they don't really know exactly how they're different, if they feel any difference at all, they will identify that, yeah, there's something there. And instead of giving up random answers, their answers will tend to one or the other, showing that indeed they can make a difference, even if that's subliminal. Perfect. So I tried this, and we figure out that it's really hard. It's really hard because subjects can tell the difference. And I tried many different kinds of arrangements of electrical simulation. I tried pairs, triples, big area. I tried to put the electrodes far apart so we can create an electrical field that will recreate the broad sensation and also the muscle twitch, everything. They could still tell the difference. Even if they, in the visual analog scale, they will mark the same things for both. In the end, they could very accurately tell which one's which. And as a subject myself, I confirm it. It is different. It's a thing. It's a taste. It's a je ne sais quoi you can identify both. And the thing is, if you can tell them apart, it is possible that the response might be different, meaning that if you're going to compare the response from the two, you'll still see a difference that is not due to direct cortical activation. You'll see a difference that is due to different salience perception in the EEG response. How much is that meaningful for TMS EEG? I don't know, but in theory, there is this possibility. So, fail. It's not possible to create a truly realistic sham using these standards. So I give up and went home and forgot about this. Then I came back the next day and I thought, okay, I have to figure out a way out of this. So what we did might be controversial, but we did the following. We tried going the other way. Instead of pairing them, we just accepted that it's impossible to pair the two. But what we can do is use a somatic simulation that is so high that whichever difference that the addition of the real TMS might do, it will be meaningless. Using the idea of the power law in the psychophysical power law, which is kind of saturating sensory inputs. The idea is that if there are two stimuli that differ in intensity, you can perceive this intensity, but the same difference, if it's in a different offset, if it's two very high intensity, but even though it's the same difference, the perceived and the response from it will be much smaller. And we can kind of intuitively think about that with weights. If you hold two bags, one weight 2 kilograms and the other 1 kilogram, you clearly know which one is which. But if you take two bags and one weighs 20 kilograms and the other 21 kilograms, well, that's kind of difficult to tell them apart, even though it's the same difference, right? Well, the idea is that this kind of works for response, from this kind of response, obviously. And we can maybe use that. So if we kick up the electrical stimulation to say 26 milliamps, this number has a meaning, I will tell you later, maybe the addition of a real TMS on top of it will make no difference in the signal. And this is what it looks like when we apply this with different intensities. This is the sensory threshold. So this is auditory only with the masking noise working just fine. You see here that you can barely see anything. And if you increase the electrical simulation intensity, you start seeing this N100, P200 increasing in amplitude, but kind of plateauing between 300 and 400. And so, well, if that is the case, and we apply these two, not these two, we apply this in a TMS EEG experiment, that means that maybe we can subtract the signal from the sham from the real, thus obtaining true taps. Using that, and in the end, this is where this number comes from. 26 milliamps is the average of all the subjects, is the average 300% of the sensory threshold from our sample. And comparing the two sensory inputs, they are pretty much the same using the visual analog scale. Using the two alternative force choice, that was a bit tricky because still, even still using this, we had a subgroup of subjects that their answer was no difference from random. according to the distribution, this distribution here. But still, we had some subjects that kind of gave answers that clearly were not random. And these three guys here that, regardless of what we did, they could always tell. They could always feel the TMS behind all this electrical simulation and noise and everything. So kudos to them. And in the end, this is our result in terms of taps. You see that subtracting the real, the sham from the real, this N100, P200, it gets abolished here. So this suggests that we are maybe going the right way. And beneath it, we can see things that possibly are just are indeed the taps. Of course, this is under preparation, but still we already receive criticism. And I will share with you the criticism that we received so far. Well, this is not a truly realistic sham either, because the sensory input is higher than standard TMS. So it's not realistic because that's not real. You're just using more. And say, yeah, okay, we just change the name to something else. This is only applicable to subthreshold TMS to the M1. It does not account for motor reafferent responses, which is also a problem because, well, these things that we see here, well, these are the taps from the from subthreshold M1. So this is very limited in terms of the response that it's going to give. This is not all. This is just a proof of concept. Maybe if this is true, and let me show something that we were thinking earlier. If this is true, maybe it is possible that here what we see is when we subtract the 100, the super threshold from the subthreshold from the super threshold, maybe both these conditions had a similar sensory input, so that when I subtracted them here, what I see is the true taps. And they have the kind of the same shape that Dr. Rocky mentioned, with this negative, with this N100 here, that is not the typical N100 right in the middle. It's this lateralized right on top of the area where we stimulate it. Maybe that is the true tap, although this was not controlled for motor reference. So yeah, so you have to take this with a grain of salt. In any case, back to where we were. Another issue with this thing is that peripheral activation modulates motor output. So yeah, so we are adding a lot of sensory input here. Well, but we know that sensory input modulates the motor output, modulates the motor cortex. So in theory, it might be also changing the taps. So what we see by using this method is not the real taps, it's tap being modulated by the sensory input, which might be true. But then again, all TMS EG will inevitably be contaminated by sensory input because okay, auditory masking noise, it works, it's a great strategy, but there is no such alternative for the somatosensory stimulus. How can you block the sensory stimulus if you're creating this huge magnetic field in the cortex? There is no way around it. So inevitably, unless you do some invasive stimulation, there is no way around it. You always have some modulation of the response by sensory input. You can minimize it, but maybe never truly abolish it. And finally, this saturation idea, it's only valid for a particular intensity window, because it's not really saturating. You're just approximating it to a level where it makes no difference within this intensity. But if you go to higher intensities, a big enough difference might still be perceivable. And we saw this when we tried to apply this to cerebellar stimulation. Now, cerebellar stimulation is complicated because it's a kick to the back of your head. It's very uncomfortable to try to actually reach the cerebellum with TMS. You'll get a monstrous motor response because you're also stimulating the trapezium. So we tried to figure out this optimized sham here. We use high electrical stimulation with electrodes in the here in this region here. Still, it wasn't enough. So on top of that, we decided to use a second coil that would also produce, obviously, the click on top of the shoulder and the masking noise. Still, when comparing the cerebellum to the sham, we still see this N100 here that is very similar to the N100 that we see when we stimulate the trapezius muscle. So yeah, super tough. Interesting, and in the time frequency, this can be translated into this massive alpha desynchronization here that you also see here, suggesting that indeed this was, even using all that, that was not enough to saturate the sensory input from the cerebellar stimulation. However, because this is a control, we control this with occipital stimulation because, well, maybe whatever response that we're seeing here is occipital stimulation, stimulation of the occipital cortex. Interestingly enough, when we, in the occipital stimulation condition, this was not the case. So apparently this was enough indeed to control for the occipital stimulation, because you see the N100 here, it's completely abolished. And we only see this difference here, which I'm not showing here, but I guarantee this peak here is in the occipital cortex. It's most likely the product of the direct stimulation of that cortical region. So the point is, there is no one size fits all. This saturation thing, it has to be controlled for every different experimental setup that we're doing. Still, it might be a promising area to be explored. And possible future directions from here. Well, maybe we should try to figure out a way to control for this motor reafferent potential if we are using suprathreshold stimulation of the M1. All this method is super nice, but maybe it's an overkill. Maybe it is. I just aimed super high, but maybe it is too high. How similar or indistinguishable real and sham must be for a proper control? Maybe it is not. Maybe the saturation thing is not really necessary. Maybe just making sure that the subjective perception from the individual, from the real and the sham, are similar. Maybe that's enough. I don't know. But if it is, it will make our life easier. We wouldn't have to rely on this complicated setup. But this needs to be defined. And to what extent does the peripheral activation modulate the TAPs? I don't know. This is a theory. This is in theory possible, but needs to be explored if we end up having to use this optimized sham. And of course, given that this peripheral activation is actually a big deal in TMS EEG, what about the previous studies showing modulation of TAPs that I've shown in the beginning? All these pharmacological experiments using TMS EEG as a disease marker that's shown differences in the N100. Well, are those real TMS EEG differences, or the differences in the N100 that they've shown, they just represent modulation of event-related potentials that could have been acquired just by using noise stimulus or somatosensory stimulus? That, that should be investigated. And I would be very interested to know to what extent these are indeed modulation of TMS-EEG potentials, or simply, peripheral activation potentials. So that is it from my part.
Okay, fantastic. Well, thank you so much for that. That's fascinating, and the sham control is very interesting. I look forward to hearing people's questions about it. We'll move right along because I think we're running a bit behind schedule. And so now I'll invite our final speaker. And so Sina Shinpur, if you don't mind, I'll invite you to present.
Can you hear me?
Yes.
Yep.
Great. Okay, hello everyone. My name is Sina Shirinpour. I'm a postdoc in the OPITS lab at the University of Minnesota. I thank the organizers for inviting me to give my presentation today. And today we had two great talks about TMS EEG, but I'm gonna present, take a different approach to TMS EEG and present my paper from last year on using phase-dependent, closed-loop TMS EEG in real time. So you're all experts in TMS and EEG, so I skipped this introductory slides, but a couple of important things to take note when we deal with EEG is that, as you know, EEG is made up of different oscillatory oscillations, like in different frequency ranges, like beta, alpha, theta, and so on. And from previous literature, we know that each of these oscillations have a functional role in the brain, like they each are involved with different brain activities. And if you look at these oscillations, we can study different aspects of these oscillations. One of the things that we are interested right now, like in this paper, is looking at phase of these oscillations. And phase is basically just a temporal shift of a periodic signal. And the reason we care about phase of oscillation is that there has been some studies that reported that phase of the EEG is also important, like functionally speaking. For example, it has been reported that peaks and troughs of the alpha band denote high and low excitability of the state of the brain. But most of these studies are in offline post-hoc analysis, that they have applied everything like TMS in an open loop fashion, recorded brain activity, and then looked at the phase of the brain activity when the TMS happened, and saw that there are some differences. But because we know that phase is an important aspect of this oscillation, why not use these to inform us about the state of the brain in a closed loop fashion? And when we say closed loop, we think something like this. We can record brain activity like with EEG in real time, and then we can extract the signal from the region of interest, and then we can bandpass filter the data, let's say in the alpha band. And based on that oscillation, we can take a last few samples in the data, in a window, and detect the phase. And for example, let's say we're interested in the peak of the oscillation, we can detect the phase and then send the TMS trigger to stimulate the brain at the peak of the oscillation. But the problem is that this diagram is too simplistic. In reality, we have to deal with some challenges like causality, which I'll explain later. Also, we have to deal with system delays. We have a lot of hardware and software delays. And also we have to deal with the speed accuracy trade-off, because we want to run the whole thing in real time. It has to be fast enough. And that means we have to sacrifice some of the speed, sacrifice some of the accuracy in our analysis. And just a brief explanation of what I mean by causality limitation is that if you're familiar with signal processing, you would know that when you like filter like a chunk of data, because we don't have the data from the future, we only have the data from the past up until the current moment. When you bandpass filter the data, you will get some distortion around the edge of the data. And like this is an example, the blue signal is the ground truth that like I have filtered in a post-hoc analysis when I have the whole data. And the orange signal is the like causal filter data, like in real time. And as you can see, like around the edge of the signal, we see some distortion, and that totally throws off the phase of the signal. And because we are interested in detecting the phase of the signal in real time, exactly the region of interest is distorted. So we have to do something about this phase distortion. And the way we solve this problem is first by like getting rid of this. Let me pull up my laser pointer. We have to get rid of this distorted signal, and then we forward predict the signal based on the signal that we have. Based on the signal that we have so far, and we can easily detect the phase after we forward predict the data and then send it TMS. And basically this forward prediction is the basis for all methods that has been proposed to do phase dependent closed loop TMS EEG. And let's give a brief introduction of the previous methods that have been proposed for like phase dependent closed loop brain stimulation. And generally speaking, at the time that we were working on this project, there were two different categories of forward prediction. Forward prediction in the time domain and in frequency domain. And here I'm going to focus on one example of each of these categories. I will briefly explain each of these methods, but I will not go over the details. So the first method is autoregressive or AR model, which is a forward prediction method in the time domain. And the way it works is like this. Imagine the black signal is your raw data that you get in real time. You can bandpass filter the data, for example, in the alpha band, and get rid of the edges at the beginning and at the end, because as I said, the edges are distorted, and you will end up with the blue signal like this. And after that, you can train an autoregressive model with this equation on the blue signal that you have. And after doing that, you can like basically use the samples that you have to predict the future. And if you iteratively do that, you can predict the signal like this in the time domain. And after that, you can apply a Hilbert transform and get the phase of this signal in real time. But the thing is, the autoregressive model has a good accuracy, but it's very slow because training this autoregressive model is very computationally demanding. And it requires a lot of careful parameter tuning, which makes it hard to use. The second method that we're going to discuss is fast Fourier transform, or FFT method. And as you're familiar, FFT is a Fourier, is a frequency domain analysis. And the way we forward project the data in frequency domain is like this. Again, the black signal is our raw data. We bandpass filter the data and we get this purple signal here. Then we project the data into the frequency domain by applying Fourier transform. And then we look at the spectrum of the signal and we take the a frequency with the strongest amplitude. That basically tells us the dominant frequency in that signal. After that, because we know the dominant frequency, we can simplify this purple signal with a single oscillation, with a single frequency, with this dominant frequency. And this is our sine wave in the red. And because this sine wave is just one single frequency, we can project it easily in the future and like detect its phase easily. But as you might imagine, because we are like getting rid of all the frequency content of the purple signal and only keep one component, you are losing some information here. And because of that, the accuracy is not that good, but because the method is simpler than the autoregressive method, it is relatively fast and there are not that many parameters to tune. But because of the shortcomings in these two methods, like for FFT, the problem is poor accuracy. In AR method is that it's not easy to use. We had to try to come up with a better method to solve those shortcomings. And we proposed the third category of algorithms, which we call training-based algorithm. And unlike the previous two categories of method, in which we only use the data that we acquire in real time, in the training-based methods, before doing the real-time experiment, we record a little bit of data. We analyze the data and extract some features that we care about that would help us with phase detection. And based on those features that we have analyzed during the training, we use them to help us with predicting the phase in real time. And we proposed a simple method called educated temporal prediction in this category, or ETP for short, that help us detect the phase easily. And here is a brief summary. So as I said, we record some short data, like for example, 3 minutes of resting state EEG data before doing the real-time portion of the experiment. And then we analyze the data to extract some features. Here we looked at a very simple feature, which is inter-peak interval. Basically, we bandpass filtered our resting state data and we get this oscillation here. And we extracted, we looked at the distance between any consecutive peaks. And that gives us a bunch of intervals between each of the peaks. And if we look at the histogram of these inter-peak intervals, we end up something like this. And as expected, sometimes the peaks occur closer together and sometimes the peaks are farther away from each other. But on average, typically speaking, most of the peaks have a certain distance, very close to a value that we call T. And you can think of it as a, let's say, a cycle period of that oscillation. And after we have our value T, we can use that feature in our real-time experiment. So let's say this is their black signal is the data that we have acquired in real time. We can bandpass filter the data and we get this signal, and we get rid of the edge because as we discussed, the edge, there are distortions around the edge of the signal. And then I detect the last stable peak in the signal and just project it with the value T, because I know that typically speaking, there is a certain distance between the peaks called T. And if I have the last peak, I can easily project the next peak. And as you see here, unlike the previous methods, I don't bother with projecting the whole signal. And I only project what I care about, which in this case is the peak. And because of that, as you might imagine, this method is faster than the previous method. And I have one disclaimer for the sake of time. I just summarized all of these descriptions. And this training thing is slightly more complicated, and we do some adjustments during the training phase to find like the adjusted value of t to increase the accuracy. But regardless of how we find this value of t, the concept is still the same during the real-time portion, that we can project the last peak into the future and find the next peak. But let's, like now that we have discussed the methodological details, let's talk about how we validated and compared those methods. We had two ways to compare those methods. First of all was like simulation based on pre-recorded resting state EEGs that we found on some databases. But also we actually did in vivo experiments, like in real time, close to TMS EEG experiments as well. And this is the setup that we had. As you can see, this is our TMS machine connected to a TMS coil, and the participant sits here. We record EEG with this EEG cap. The data goes in the EEG amplifier. We stream the data in our computer where everything is analyzed. And when the right time comes, a trigger is sent to the TMS machine to stimulate the brain. And in our experiment, we did this closed loop setup in 3 different brain regions at the occipital, motor, and prefrontal cortices. And we applied all these 3 algorithms that we discussed in randomized order. And the way we like localize the data in each of these brain regions is by using a Laplacian montage on the region of interest. And the results that I will show you is focused on the alpha band that we filtered the data between 8 and 13 Hertz. And here is the results that we have for simulation. And before explaining that, let's go over a brief introduction of what these plots mean. These are called polar histograms, and you can think of them as like very similar to regular histograms, but because we care about phase, and phase is a periodic variable, instead of plotting everything in a regular histogram where things can go between -infinity to +infinity, we plot everything in a circle that goes from 0 to 360 degrees. But similar to regular histograms, each of these bars correspond to the number of values that we get in that bin. And like ideally speaking, because we want to target our desired phase as accurately as possible, we want the difference between the targeted peak and the desired peak to be close to 0 as possible. And in a polar histogram, that would mean that ideally we want all of our bars to be as like stretched towards 0 as possible. And if you get more spread, something like this, like as the more you get close to a uniform circle, that means that you are closer to chance and you have like lower accuracy. So if you do an open loop simulation, you will get like a uniform circle. And as you can see here, just visually speaking, you can like even see that our method ETP is like more stretched towards 0. That means that we have more accuracy compared to the 2 other previous methods. And ETP and AR are both better than FFT. But also we quantified the accuracy using this equation, which is nothing fancy. It's just looking at the norm of like some of the norms of the deviation between the detected phase and the desired phase. And what we see here again is that ETP is like has higher accuracy than AR, and they're both more accurate than FFT. But also we see something interesting, that in occipital cortex, regardless of the algorithm, the accuracy is higher in occipital cortex, and motor cortex is like prefrontal cortex is the lowest accuracy among all of them. And I will discuss why we see this. And that was the simulation results, but also in the experimental, like real-time actual experiment, we see the same exact pattern. ETP is more accurate than AR, and FFT is worse of them. And just to give you an idea of what I mean when I say our method is faster than the other methods is, as you can see here, each iteration of like this the analysis takes like much faster than the other two methods. And or in other words, our method is more than 10 times faster than the previous methods. And that is important because it means that you can run our method in any regular processor. You don't need fancy hardware to be able to run this in real time. And another thing we looked at, as like you intuitively expect, the higher quality data would yield you like higher signal to noise ratio, and that would mean that we can detect the oscillations more like better, and therefore detect the phase in a more accurate fashion. And our results like confirm that the higher your signal to noise ratio is, the higher accuracy you will have in phase detection. And so that was a conclusion of the paper that we published last year. And as a summary, we were able to use like EEG oscillations, the phase of the EEG oscillation as a biomarker to run closed loop brain stimulation with TMS EEG. And we compared 3 different methods and showed that our method is faster and more accurate than the previous method. And also SNR is important for accurate phase detection. And in case you're interested in this project, you can like access all of the codes for the real-time application in our GitHub page. And I will briefly explain some new things that we're currently working on as an extension of this project. So our postdoc, Miles Wisniewski, is currently working on using this technology, the closed loop technology that we have, to study physiological outcome of closed loop TMS EEG. And the way we're doing that is by applying like this closed loop TMS EEG at 4 different phases, like peak, falling phase, trough, and rising phase, at 2 different frequency bands, like the mu rhythm and beta rhythm. And we apply TMS to the motor cortex, and we measure the MEPs, like with EMG, and we like detect the finger twitches. And like our preliminary results show that indeed not only different phases have an effect on how like the brain excitability, but also different frequency band has an effect. Like we see that in mu rhythm and beta rhythm, we get a flipped like signal. And this is like exciting work that we hope to like submit soon. And this shows that the closed loop application actually works, and it has an effect on the brain. Also, one last thing, we are currently working on like a new algorithm. It's currently work on progress, so I don't have any results, but we are trying to improve the ETP method even further and be able to run a wide range of frequencies, like look at theta, alpha, beta, and even gamma, like oscillations, like in a robust fashion to be have a more general usage of this closed loop TMS EEG algorithms. And I want to acknowledge everyone who's been working on this project in the OPITS lab, and also funding agencies for providing the money that let us do these projects. And with that, I want to thank everyone for listening to me, and I would be happy to take any questions.
Right, fantastic. Well, thank you so much. That was very interesting. It sounds like the beginning of almost a new area of research. So I'd like to thank all the speakers for being for giving great and fantastic talks and for being very generous with their time. I know that we've run a lot more overtime than we expected. So I don't want to impose too much more on the speakers, but we might ask a couple of questions if that's okay, as long as it's not too much of an additional imposition. So I might just have a quick question for you, Sina, if you don't mind. I mean, so I know you're expanding into additional frequencies. Have you also thought about expanding into not just resting state, but potentially task-based uses of this closed loop system? So, I mean, I know you can't do tasks that have a sort of definitive stimulus because, you know, it's essentially just triggering an ERP, but possibly tasks that might have a more extended design, you know, maintenance tasks, for example, or something along those lines. Is that something that you've considered?
Yeah, definitely. That's an interesting thing, and we have thought about it. We're starting to explore that, but I can only speculate right now. And if you have any task that, as you said, has like those prolonged effects, like on oscillation, as long as your oscillations are stable, it really doesn't matter if it is a resting state or any task-based like activity. But if you're talking about tasks that induce acute like changes in the brain response, then that would be a tricky scenario, and that would be much more challenging to do this because like brain activities would suddenly change, and it would be more challenging to track those phases and deliver those stimulations. But for the prolonged, like stable oscillations, we can definitely try to do closed loop stimulation.
No, I think it'd be really interesting to see. Yeah. So we had a few questions in the chat as well. I think there's a lot of interest to do with the auditory evoked potential, and you know, quite a few asking whether all along the same sort of theme, asking whether you recommend using insert earphones to present the masking noise. So I imagine insert earphones as opposed to headphones, the use of foam to reduce the TMS clicks sensation, and I'd be interested to hear whether there's a difference of opinion. Is it such an important? Should we accept that the AEP, the auditory evoked potential, is there, and you know, focus elsewhere in the signal for what we think is the true TEP, or you know, do we need to spend a lot more time trying to dial down the auditory evoked potential to get at the true signal?
Shall I begin? Who's taking that?
I'll leave it to both of you to answer.
Okay.
See what
so with regards to the form, I think the first thing you asked, right? I'm not sure about whether it's really necessary or not. I mean, I tend to use it because it has been reported that those auditory components are larger if you don't use the form. That's where the idea of bone conduction of the TMS click, the vibration came from. To be honest, I didn't see much difference in my experience, but it doesn't really cost anything to put a very tiny layer. So why not? I mean, I would advise to do so. In terms of should we accept that the AEP is there, the answer is no. I don't think we should. And I don't know. My experience is that at least in the intensity I explored, which was sub-threshold, maybe I don't know whether it was clear or not, but in my paper I used 90% RNT. Okay, so it was not a very big intensity. The AEP was suppressed effectively, and this was reached with two methods. One was using masking noise with earphones like this, and then putting ear defenders that I think they were cutting like 30 or 35 decibels and so on. So it's two, one on top of the other. I think the second does a lot of difference, and it's really cheap, and it's really, you know, easy to implement. You just have to put it here and put the band below the chin. So that doesn't cost much, and it does a big difference. So I don't think we should just accept that the AP is there. No, I think it can be suppressed within a certain intensity range. Then if you ask me what happens if you just turn the stimulator to 100%, is it possible to suppress it completely? I don't know. That might be trickier. But at least for subthreshold intensities, I think we should aim for a complete suppression. Then, of course, it's also a question of subjective tolerance. I mean, I saw in the chat someone was asking whether what do you do if if people don't tolerate it? Not much, not much. That's a problem. But at least with healthy subjects, that can tolerate quite a bit in the lab, I think we should reach a point where the AP is suppressed.
Okay. And I guess I've got one question as well for you, Dr. Gordon. So with the design, a very interesting design for the sham control, I think it's fascinating. And so you'd opted to increase the saturation of the electrical stimulus. So you dialed up the electrical stimulus and sort of tried to level out the difference between the sham and the active. Have you thought about almost approaching it from the opposite end, or maybe in conjunction with this second idea of using something like a topical analgesic to numb the sensation? You potentially doing both simultaneously, numb people, but also saturate their numbness.
Yes, that was one of our ideas, like our bench ideas before we actually started the study. So I was, oh, why don't we do this? And the thing is the following: topical anesthetics, like in form of a cream, they are very inefficient to actually. They're inefficient. They don't actually work. If period. You have to actually inject subcutaneously some lidocaine or some other sort of anesthetic. It has to be in a large area. You have to consider whether if it's in a, well, if it's right in the midline, yeah, maybe it's okay because it's only going to be stimulation here. But if it's lateral, then you get the muscles, your temporal muscles or your frontal muscles. Then you have to worry about not only controlling for this sensation, but also maybe blocking these muscles with anesthetics, so you get another layer of complication. Then some people say, well, when you're doing this, when you're doing the TMS, you also stimulate the neural terminals in the dura, and maybe those also need to be controlled for, and then how you're gonna anesthetize that region is, well, then whatever. Let's just forget about it and go the other way. Because also, even if you're just going to with the local subcutaneous anesthetics, that's kind of an invasive procedure, and yeah, that definitely makes it more difficult, one, to get through ethics committee, because we're doing a kind of unnecessary procedure in healthy subjects, so that's certainly complicated. Second, it's going to be harder to get participants. So yeah, so then we go, okay, no, it's not a very good idea. And then you might say, well,
basket.
Yeah, well, so then why don't you go do an invasive procedure like a transcranial electrode that goes inside the cortex? But then again, that's not the purpose of non-invasive stimulation, is it? So yeah, until we figure out a way to non-invasively stimulate the cortex without all these sensory inputs, yeah, we're stuck with these issues.
Okay. Well, hopefully with some more work and by all of you, we'll get closer and closer to a solution in the coming years. We do have a few more questions in the chat, but I think in the interest of time, we've gone more than half an hour beyond what we initially said was going to be the finishing point of the talk. So I imagine everybody can read the articles that you presented online, and they can, the corresponding authors are there as well. So I'm sure some of these questions might be answered by those means. So I'd just like to thank all of you again for presenting. Thank you so much. It was fascinating. It's very interesting. Really enjoyed listening to it. Thanks to everybody for attending. And if you would like to see the video recordings of this this presentation, we'll be putting that up soon. So thank you all, and good morning, good evening, good night, good afternoon, good whatever time difference you're at. Until next time, thank you very much. Bye, everyone.
Thank you. Bye-bye.
Bye.
Bye, everyone.